Okay, so the next part of the Lodaat masterclass is on innovation and technological advances in manufacturing. And we thought we'd do something a little different. We thought we'd have a panel discussion, and we've brought three of our scientists for the panel discussion, and we really recommend any questions. We want to make it a little bit more interactive. I'm going to be asking the questions as moderator, but you know, please do chime in, and if you have any subsequent questions to the answers, please raise your hand, and you know, we'd be more than happy to try to make this a little bit more interactive, if you will. So just again, very briefly, Lodaat, live one day at a time. And we are here to answer not only any questions, but also to provide information on generic pharmaceuticals, nutraceuticals, dossier and tech transfers, turnkey solutions, and API ingredients. And we are going to be asking questions specifically on these and specific for natural extracts. So I want to introduce the scientists that we have. I'm Rajiv Khatau, by the way, if I hadn't introduced myself. You had just met Dr. Ranjan Mogre, who is our Chief Science Officer. And Dr. Mogre is very well known in the industry. We're very lucky to have him as part of our team. He has a PhD in bioorganic chemistry from Boston University and over 40+ years in the manufacturing processes. Dr. Bhate is also here. He is here from India, and he's a leading expert on analytical chemistry from the University of Mumbai. And Dr. Vaidya, who's also here as well, has over 10+ years and also on the analytical chemistry. So if you guys can come on over. We probably should have introduced everyone by how they're being seated. But we've got Dr. Mogre, Dr. Bhate, and Dr. Vaidya over here. So the theme of the lecture is what new technologies to aid in the dispersion of hydrophobic compounds. We also want to focus on nanotechnology and nanoencapsulation as possibly one of the most promising technology trends, and also how is continuous investment in innovation and R&D occurring, and specifically what are the tools that are available to manufacturers, both to strengthen the technology as well as on clinical innovation. So hopefully we're going to get that out of this discussion. So with that, I'm going to start off with one of the first questions, and you guys chime in. So I'm going to keep it open-ended and just ask in terms of what new technologies are out there, and specifically on new formulation development, as well as any other technologies that may actually exist. What's sort of the cutting edge, and what's maybe not so cutting edge? And I'll let...
This is on.
Yeah, it's on.
See, there are two different parts, as I understand from the question, is what are the new technologies coming in. One is to make the API, whether it's natural or synthetic. Second is to make the API relevant for the formulation one want to develop it. Second is what's the new technology in terms of the delivery systems that would be happening. In terms of the availability of the APIs or the manufacturing of APIs, I believe we are probably reaching to a saturation level of, you know, developing new technologies for the extraction, developing new technologies for the chemistry. What one actually would be doing, you know, even further down the line, would be only utilizing the known techniques to modify to the requirements of the applications, you know. But mainly the technology in terms of extraction, as far as the supercritical extraction, probably is the latest now, and probably would stay there for a while. There would be different solvents one would use in supercritical extraction rather than carbon dioxide. There would be nitrogen as the cryo extraction techniques one would use it. in terms of the other synthetic chemistries, known chemistries probably won't change, but the purification of the API, once you do the synthesis, probably there would be more changes in the purification techniques and the particle sizing of the product, of the API. That's the micronization or nanoparticles that would change in it. In terms of the delivery systems, again, there is no end or no limit that would be there. We had the very simple solid oral doses earlier, the tablets, the capsules. Now we have, the pellets, the tablets in capsules kind of thing. Then now we have the nano particleized material or micronized material in tablets and capsules. You also have the injections, and you also have now the liposome-based delivery systems, which is getting more popularized into it, mainly used for the biological molecules or biomolecules or biosimilars. We all know that these are based on the liposomal drug delivery systems. Like the main application, as I understand, is you work with anti-cancer products, which like RNAis. These products actually really work on anti-cancer applications, but the limitations are happening because how we actually deliver the RNA dye to the site, and that's happening with the liposomal delivery systems.
Ranjan, in continuation to this question, I would like to ask you one thing about the nanoparticle size, what you were talking about right now. How to what level you can reduce the particle size of API, whether natural or whether synthetic, by using supercritical fluid extract?
Yeah, it's an interesting technique. People so far have been using it for extraction of the raw materials, so whether you want to use it as a perfumery products or the medicinal application. But recently, it is really got into a challenging application of micronization. So far, we were known to, a relatively not correct term, a destructive or a heating technique or beating technique, like you actually beat the powder and make it smaller particle size, or even jet milling, which would give you a good micronized API from, I would say, up to a range of 5 micron or 1 micron sometimes. But there was a danger that you would lose the morphological structure of the API. In recent years, what the supercritical micronization has done is, it has reduced the particle size from 1 to 2 micron to submicron level. To my knowledge, I recollect people have succeeded in taking the particle size to 0.2 micron, which is about 200 nanometers. This is actually potentially very useful in terms of the product, which people would try to use it in terms of the aerosolic applications, where you need a fine API so that you can make the cloud of the drug much easier.
Any idea about the advantage of this supercritical methodology for particle size reduction, particularly in the case of APIs where agglomeration is observed in the common methods?
Yeah, the agglomeration is to a larger extent is taken care of in the supercritical micronization technique, mainly because the technique really works like you have the product either in carbon dioxide or your product in the most preferred solvent, and then you actually start precipitating with the two solvent coming down from two different nozzles. And that actually limits the agglomeration of the two particles. It also limits the filamentation of the APIs. The more common application of this would be, small molecules of polymeric natures, like the peptides kind of thing, where you need the peptides otherwise are very big molecules compared to a generic molecule, and you really want it to make a smaller particle size. So that would be an area of interest.
So the yes, please.
My question is to you guys. Going through the exhibition here, and I see numerous vendors or the exhibitors are showing API products here. And everyone is so much of the API. I mean, there are thousands of them or hundreds of them. What is the difference between this API? Why is it that business is so lucrative, or what's the reason for that? And I don't understand that. Is anyone can explain to me?
You want?
With the micronization, what are you asking?
No. There are more.
I think to summarize the question is the differences in API per se, and how the competitive advantages within different firms. You have thousands of firms out there, you know, whether it's in the manufacturing process or other processes. I mean, is there anything specifically that you could, you know, answer on that?
You want to answer? Yeah.
This probably relates to the leader or the patent holder and subsequent conversion of that into an open market, in which I remember one very old example, ibuprofen, which is moiety of boots. They used to make by aldehyde route, but today ibuprofen nobody makes by aldehyde route. They make by diol route, and that is much simpler to make, faster to make, and economical as well. These many companies, till the patent is on, they do try to make with different way, so that they can file different route, but efficacy, purity, polymorphism, everything has to be maintained. Now, why so many companies? Because each company has its own way of making materials, and they try to economize, and that is the basic reason. Why so many? Once, say, I make a submission and I have a contract, I file in my papers for formulation with A API, A source API. If my going into the formulation, if say Ranjan wants to make another formulation of the same, he may use B API or C API or D API.
It's going to be very difficult.
It would be
to use two different sources of API and make
For
the same
one,
formulation
yes.
and the dossier is going to be a hell of a job for any organization. So obviously the API members, once you shortlist one particular or approve one particular DMF holder, you have to stick to them. And if you want to change, you have to change everything.
complete dossier.
That becomes difficult. So that's why there are so many players in API nowadays. Again started increasing.
I want to ask.
Does it
yeah,
answer
go ahead.
your?
Does that answer your question? I wanted to talk a little bit about some of the other innovations in terms of equipment. Any of the newest types of equipment that maybe we're using or that's out there in the marketplace. I mean, is there anything that's out there that's sort of new or innovative at all?
With respect to analytical sciences, in last 2 decades, there have been many changes. Sticking to the needs of regulator, one needs to audit trail all the analysis, right from receipt of the material till its final certification. Now, when you do that, previously we used to use various manual ways, the actual chemistry ways, but today we use mostly all instrumental methods. And even in the instrumentation, like HPLC, which we saw, that was a difference of about hardly 4 or 5 years. We used to do a big analysis time exercise to achieve to the same result. But today we can do that much faster and with same accuracy, with same, what we say, resolution patterns. How did it help? Imagine you have to certify, say one typical example of solid dosage form, a tablet. You need to do purity, you need to do efficacy, that means strength, you need to do related substances, you need to do content uniformity if doses are small, you need to do even dissolution profiles in cases of delayed release or modified release. Now, for one batch release, you need to do analysis multiple times and multiple samples. So these all samples to be completed within a day used to be very difficult, but with new technologies like HPLC, you can do analysis in much lesser time, about 4 minutes maximum. And you can finish all the analysis within 1 working day. And that actually helps to release your bond room sizes also. So that material can be available quickly, releases fast, and goes to market also quickly. So it works out economically as well as more efficiently. Like these, analytical techniques have kept on evolving. Now, polymorphism or even dissolution profiles or even estimation in online using of NIR technique for the checking process, all these have really helped instrumentation-wise. You wish to add in modernization, you were adding something?
Yeah. So, we talked about a little bit about product quality. In terms of any new requirements that may be out there for product quality, if any of you want to take a stab at that, maybe Dr. Vaidya?
Yeah. You all may be aware of the specific tests as per the monograph and maybe in-house tests which have been asked by the regulators while submitting the dossiers of the finished product. And in recent years, if we look at, there is one additional test requirement came up with extractable and leachable studies, which is absolutely product dependent, packaging material dependent, and there are no specific guidelines. Only one PQRI group gives some suggestions on it, and they are only suggestions and only the concept. So it is left to the analytical chemist to design the protocol, which I think we do very well, quite well experienced with the two scientists sitting besides me in our lab at Analytical Solutions in India. We are doing a lot of these studies, and we have developed some kind of a protocol which will cover the complete requirement of the regulators with respect to what kind of compounds you are going to look at in these extractable and leachable studies. And this particular requirement is, there is a need of a lot of sensitive equipments like GC-MS, GC electron capture detector, ion chromatography, LC-MS, and so on. I mean, there is no limitation to it. Probably there will be a limitation to the existing analytical techniques with this requirement, and right now there are clashes going on with the regulators and the sponsors or the file for the dossier submission people. All the companies, they are saying you can't go below this particular limit, and these are certain challenges in the analytical field right now, which we may come out, but when? It all depends on the instrument development companies.
So in the morning session, we talked a little bit about some of the market opportunities and some of the trends that were going on. And I think Dr. Mogre briefly mentioned curcuminoids and the analysis of curcuminoids. That happens to be one of the products that's doing very well, as we all know. Is there anything else you can talk about in terms of that ratio and how not only how to maintain that ratio, but also how that particular ratio can be used for different indications.
See, there are a couple of things which helps you maintain that ratio. One is to procure the correct age turmeric rhizomes. The rhizomes, actually, the growth of these chemical components is actually a time-wise evolution. You have the curcumin, the dimethoxycurcumin and bis dimethoxycurcumin, they don't actually develop in the rhizome simultaneously. And as the rhizome starts maturing, the component ratio is more or less, comes to a fixed ratio. But depending on the method you use it for the extraction, you would preferably extract one against the other. If you do that, sometimes you end up the curcumin, which is normally required in any formulation, to a tune of about 85% plus, and the other two components totaling between 10 and 15%. Any organic solvent extraction, you end up with curcumin, which is almost like 60-65%, and other two are almost like 30-35%, which would actually bring down the efficacy of the product, because in the curcuminoids, the curcumin component is the most effective component and not the other two. But it's very difficult to separate the three from each other. In terms of the economics of it, if you leave few percentage of it, they are not going to harm your product. But they can, if their percentage of that is more, it would start bringing down the efficacy of the end product or formulation what you have. So you have to see that you have the correct matured rhizomes to pick for the extraction, and then the correct technique to use for the extraction.
Yeah, go ahead.
Just as the topic was curcuminoids, see, in India, our nannies used to make creams, skin creams for kids and everybody. They used to take butter and used to take actual roots, the dried, and used to grind on a stone. And they used to make those creams, and they used to be pretty efficacious. What he probably is maintaining, once you extract differently, and if you try to make same way, same composition, extract added and the naturally powdered buttered cream, it might efficacy-wise work differently. Yeah. Is this what you mean?
Yeah.
Have you seen?
Gentlemen, you brought up a very good question, the topic about the kokamin, and I'm all for it all my life. And is it sound enough? Okay. I'm one of the therapists, and I use this one on my patients and everyone. And every time I go and I see there's a 500 milligram curcumin on a bottle, or 700 milligram, or even, does it mean that it can be, what will be the difference between 500 and 700? I'm not a technical person, but can you explain what would be the right way? Well, it should be mixed. When you say curcumin, it has to be mixed with something, or is it combined with other ingredients, anything?
No, it actually all depends on what your end use going to be. If you're going to use the curcuminoids as an antioxidant, you should use the curcumin as an isolated component, indicating more than 85-90% curcumin in the extract you have. 500 milligram of curcuminoid, whether it's curcumin, bismethoxy or something, is actually a huge amount of turmeric. Because if you convert an average yield of about 2 to 4% of curcuminoids present in the turmeric, what it actually reflects is, you take 100 grams of turmeric rhizomes to 2 to 4 grams of the curcuminoids. So 500 milligram of turmeric curcumin in your formulation, whether you take it as a capsule or take it as a lotion or something, it's a big number. I actually couldn't get exactly what you meant of 500 milligram and 700 milligram ratio, but I imagine it's the quantity of curcumin in the capsule you're probably trying to look into. Yes, 700 milligram is obviously preferred depending on the area of application of the curcumin.
That leads to another sort of question. There's a lot of uses for extracts, obviously.
Yeah.
You've got nutraceuticals. You had mentioned that. You also have CPG firms who want to use it in ingredients, as well as in cosmeceuticals.
Yeah.
Is there, and maybe there isn't, but is there any difference in the types of ingredients, both on the manufacturing side as well as on the analytical side, that you should look for these particular different customers and their particular wants and needs?
Again, as I said, as a formulator of any finished drug product, my first reaction would be that I maintain the composition of my formulation, and that is decided by the quality of the extract I buy from vendor 1 to vendor 5. If I keep the specification of my extract rigidly fixed between the window, it would make my life more easier in terms of maintaining a consistency in my formulation. It all depends on formulation. Like I want to use curcumin as an antioxidant. I can always add that with an enhancer, like I mentioned, the Piperin extract, which makes the bioavailability of the curcumin to the body much more than the curcumin in isolation. So it all depends on
and
what
maybe,
you want to use.
and I know Dr. Mogre, we know about the Piperin, but maybe you can explain a little bit more about how that bioavailability with the Piperin works.
I know how it works, but I'm not too sure whether I can discuss too much on those here, because it's patented by one of the stallholders upstairs. So.
Okay.
But it works like, actually, if you are a biological student, you would, or even a chemistry student, you would know something like there are vitamins and there are covitamins, and there are enzymes and there are coenzymes. The Piperin, the coenzymes, what it does is actually makes the enzyme activity much more by either tagging to the enzyme or tagging to the substrate. What Piperin apparently does is actually takes the curcumin more back in the system. How it does is it's a patented aspect, so I may not be able to discuss that in detail.
No worries.
You want to explain the opening of
No, it's okay.
No,
that's fine. Current treatment of injectables and the natural way of injectables. One area may be anti-molecular diseases, and how current treatment of injections can possibly be done in a natural way. I don't know if maybe you want to... And if you don't know, we're referring in this instance...
I'm getting worried. Raju probably went through my presentation very seriously. He's probably directing to the lutein
yes, right.
extract. It's a Marigold extract, more commonly earlier used as an antioxidant, as an nutraceutical product. We all probably know the AMD is age-related macular degeneration, which is probably an order which normally happens with the prolonged diabetic conditions. You start losing your eyesight, and then at the stage you have only one option left is take an injection, which is very expensive, very painful. It's an oil-based injection, so you all know that the pains of an oil-based injection. The cost used to be, I don't remember the exact number, but it's a very expensive injection. Now what has happened with the high quality lutein extraction done by the supercritical fluid extraction technique, there are products now in the market in the oral capsule forms, which one can take it more conveniently, and that really helps. So what only one need to take care of it is the proper stable formulation. You have to develop that in a capsule form, and that's again a patented technique of formulation lutein into a capsule formulation. But that's helping. A lot of people are taking those products now, and they have prolonged the application of the injections of lutein based injections. But that's working.
In a more general way, is it possible, or do you think you can term extracts as an API? Dr. Bhate or Dr. Bedi?
Yes, I can only make one statement. Yes, all extracts are APIs. If you consider the definition of API, it is active pharmaceutical ingredient.
That's right.
It doesn't mention whether it comes from origin A or origin B. If you are going to use a natural extract as a pharmaceutical component, yes, the extracts are APIs. You have to specify the quality of the extract so that you know that API is of what efficacy.
The quality attributes with respect to extracts to be called as API will have to be very, very stringent.
Yes.
The composition, most of the times probably,
Yes.
and that has to be absolutely in a very short range of the acceptance criteria for each component of that composition, and as well the impurity profile has to be in line with the synthetic APIs which are present in today's context. Only then it can be accepted as a API. The regulators are still firm on their views with respect to this angle. They have not yet changed with respect to the natural extract to be lenient with respect to the acceptance criteria as no. That has to be absolutely clear.
There should be no differentiation whether it is natural or synthetic. The moment it falls in the category of API, the product has to fall in line with the specifications of API requirements, whether it comes from a test tube experimentation or it comes from the natural origin.
Actually, I would like to add, Chinese pharmacopoeia has many extracts which are registered, and those are all single herb extracts. Same way, USP also has taken a lead, and they have also brought in many herbal extracts, new herbal pharmacopoeia they are developing, and there those standards would be there. So if we meet SFE or SCFE extracts meet that standard well, there would be generalized material available for formulators to formulate that, and there is no harm in using that as an API.
Yes, please.
My question is related to the manufacturing technique of the herbal products. As Sir rightly mentioned.
Can you just say your name and
my name is Narendra. I'm working for Mitsubishi Chemical. So as you told us that you are using HPLC as an analytical tool to separate one compound from impurities or maybe some. So the people who knows about the chromatography, you might be using either silica for the columns. Can you share some more products where, apart from silica, that you are using for the separation, as a separation tool in the chromatography?
In fact, those were separated on reverse phase HPLC. Sorry, we did not give all those details. We thought we would run out, and hence we did not elaborate on those. And anyway, his lecture was not related to analytical, so he did not tell. But these were done on reverse phase HPLC. Of course, the first 26 minutes run was by standard 5 micron C18 column, while the second one was latest technology of the fast particles. That means they have very small silica in between and porous shell outside. I wanted to avoid branding, but there are Sigma, Ulrich, they give those columns. Supelco gives that column. Thermo gives the columns. Accent or Accucore or those technologies.
From Thermo.
This was run on Accucore, and that's why if you see the flow rate was 1 ml per minute, which used normal HPLC and still achieved analysis within 4 minutes. If you could see the resolution and even the impurities were getting separated exactly similarly. However, same thing could have been done with UPLC also. But this avoids this new technology of columns, basically, that gave us this advantage. Same HPLC could be used with, so that is.
But HPLC is generally used for analysis. But our main aim is to commercialize the product. Okay. So when we go for the commercialize on a big, when we scale up the whole project, so then what material we should use? Because silica, we all know, have certain problems.
You're asking in terms of the preparative scale?
Yes,
preparative scale you are asking in terms of probably?
The commercialization.
Right. In that case, you have to go for a preparative scale. There is no other choice.
See.
Which material? Like silica, we all know that is a problem, pressure problem, then you cannot use in the alkaline solution. So what other materials we have for that?
See, there are a combination of techniques one should use it. You should use liquid chromatography, whether you're using the silica based or alumina based or modified silica based solid support. There is something called flash chromatography. I'm pretty sure you must be familiar with it. Using HPLC alone as the technique to purify kilograms and tons of material would be very expensive. The cost of your product would be exorbitantly high. It's expected that a chemist normally work on a conventional chromatographic techniques like the flash chromatography, where you can finish separating the components much faster. You purify to a reasonable level, and that small quantity you finally put it on the prep HPLCs. Whether it all depends on the molecule what you want to work with and the sensitivity of that molecule to the solvent you would be using to separate it from the solid phase. Then that decides whether you want to work on common silica column or modified silica column like the reverse phase ones, or even higher modified columns like the amine columns, depending on the molecule you want to separate it. But it is expected that you first work with the medium pressure techniques like the flash.
Does that answer your question? Yeah? Okay. In terms of, some of our customers are CPG firms, and I don't know if there's any CPG firms in the audience here, but we get asked this question a lot about taste masking. Is there anything in particular that you can comment on neutralizing taste, especially in, hot selling products like maybe curcuminoids or some other masking?
Some formulations you're
See, the taste masking these days is more common. Whether you talk about taste masking of a natural product, the simplest product we talk about erythromycin or even ibuprofen, for example. If you just take that product as such, it's going to be bitter and completely non-palatable. So you need to mask it by something which really makes it more palatable, and that is same for all products, whether it's curcumin or ibuprofen. And that's the art of a formulator. It's more than the science of a formulator, you know, because everyone knows, you know, these are the test masking agents and these are the products where you need to test mask it. The formulator skill actually comes into picture that he masks the test, but at the same time makes the molecule available for the efficacy without even, you know, blocking it or degrading it to an extent. So these techniques are known now. There's nothing rocket
in
science
terms of
about
for natural extracts and APIs, you'd say that both of them are exactly
both of them
pretty
are
much
same.
identical in
Yes,
terms of that.
because there is no basic difference when it comes to the medicinal activity of the component, whether it comes from the natural source or it comes, as long as the chemical or the physiochemical properties of the molecule remains the same.
When you say it is a pure extract, it is almost pure like a pure compound. So it has to behave with the chemistry is the chemistry, it has to remain same. You can use the same technology what you are using right now for APIs, the same way you can apply it to this.
So, and we're running almost out of time, so we just have a couple more questions here. In terms of technology transfers, maybe we can talk a little bit about technology transfers. What are some challenges that are currently occurring? What are some trends that are occurring? Dr. Vaidya, do you want to?
That's the main crux when you go for the scale up, and it remains same as those of small molecules, even in case of natural product extracts as well. You have to make a technology transfer with respect to the manufacturing process as well as analytical methodology for testing, both, and both with the validated processes. That's the requirement as far as regulatory part is concerned. And apart from the regulatory part, I would say it's always better to do this in terms of as the R&D knows what hurdles they have crossed during the development, and the manufacturing or the production and the QC, they do not have time to look at those hurdles. So they need a straightforward solution when they are going ahead for the scale up. Of course, there are some issues when there are scale-ups and during the scale-up time, but then those can be overcome very well if during R&D, during product development process and during analytical development process, you have already studied the variations that can occur in the product. Then that can be easy. But then it is always better to do this in a proper way so that you can manufacture the product batch to batch with a consistent quality.
So the issue in terms of the tech transfer is actually taken care of these days. What are the hurdles that would come into tech transfer at the formulation development stage itself? You know, there is a more common term these days is known as DOE, design of experiments, or the quality-based designs of the formulation or the finished product. If you keep all those aspects in the mind when you are developing the product itself at that stage, with a complete validation of the analytical techniques used for the analysis of the product under development, keeping in mind what are the machineries available at the manufacturing or the scale-up side, if you keep all those aspects in the mind and then develop the product, it becomes much more simpler for the tech transfer to happen. If the product development happens at a place which is done with the set of machineries which are not at the manufacturing side, then you start getting into the more difficulties and problematic areas. Then you again go and start reinventing the process at the manufacturing side. Otherwise, it becomes a more or less straightforward job of tech transfer. We've done huge amount of tech transfer analysis for the
Were there any other questions in the audience? Well, with that, want to thank all of you, and hopefully you found this Lodaat Masterclass for Natural Extracts very informative, and thank you very much.
Thank you.
Thank you.